what do we see? What kind of results do we get? And how does it help with some of the current questions that we're looking at? So one of the things I mentioned is we can irradiate one cell, we look at its neighbors. We found something called the bystander response. So what happens is you irradiate one cell in a culture, and a whole bunch of them act like they got hit too. It's like standing in the middle of a crowded bus stop. Somebody has a heart attack and 5 people fall down with heart attack symptoms. It's called the bystander response. Again, it's a response that we're seeing near to a radiated cell, but in cells that were not irradiated themselves. They have no energy deposition, but they're responding as if they got irradiated. The cells are talking to each other. And we've seen this. I mean, and microbeams are one of the best ways to do this. We can guarantee we hit that cell and not that one. And so we can actually go through and parse this out. Here's a classic example. We irradiated these blue cells. You'll notice, and we didn't irradiate the red ones, but you'll notice there's these little spots right next to the nucleon. When the cell went to divide, there was damaged DNA. It didn't know what to do with that, so it spit it out in a little micronucleus. So these are DNA damage fragments that get spit out, and it's actually a response that we see, and we can actually measure dose from looking at how many micronuclei we are. But this cell didn't get irradiated, but it's showing DNA damage. The bystander response. The cells are talking to each other. So what does this come into? Well, it kind of concerns low dose risk. You go back to the radon story. You inhale radon, one cell gets irradiated, what happens to the tissue around it? How do you estimate the risk? From some of the data from 2001 we're looking at, what percentage, how many of the cells in your culture get irradiated? If you have 100% of your cells that get irradiated, you get a lot of them that show mutations. Okay, they're all getting radiation. If you irradiate 20%, 1 in 5, the response actually doesn't go down that much. You still get a lot of response through the whole population. Okay, so 1 in 5. How about 1 in 10? The response goes down a bit. 1 in 20, the response stays up like that. All of the classical models have said, well, shouldn't you extrapolate from 100% to 0? If you draw that line, you might be horribly underestimating the risk that you actually have from very low doses. Again, a low dose is only a couple cells get irradiated, not all of them. This is actually an active, ongoing debate. This paper was 14 years ago. I was at the Radiation Research Society annual meeting. It was 2 weeks ago down in Florida. I was there. We had a whole session to talk about this. How do we predict low-dose cancer risk? What's important here? Is this right? Is this important? How important is this? It's an ongoing debate, and we're still working on it. But again, looking at neighbors, this is something that we're looking at currently is how well can we watch what's going on in real time? So what we're going to see here is we're going to irradiate this cell right here across. And what I want you to do is I want you to watch the green signal in these cells. What this is a calcium reporter, and it's actually signaling between the cells. So we'll get to this. So we did the irradiation, and right about here, it starts to flash green. It's a little bit hard to see on the screen. I'll run that one again just right. Yeah, there we go. So again, in about 200 seconds, it'll flash green. And the signal comes up from the tree, and it just kind of oscillates. The thing is, if you're really observant, and I wasn't the first time, because I was kind of surprised to see this one flashing, there's a cell down here on the bottom that flashes at different times. They respond differently at different times, depending on the signaling methods they get. That's what we're working on right now, is how can we find out what the cells are saying to each other, and and what's the timing? Cancer treatment with x-rays. Again, okay, this is a standard cancer treatment right now where they use IMRT. You irradiate the patient from multiple different angles. You get a very high dose right where the tumor is. Again, use the CT scan, using their tools to actually, and in a proper way, actually find out where the tumor is. We can target it, we can irradiate the tumor 5 different directions. It saves dose from the healthy tissue, but there's still a lot of healthy tissue. The X-rays come in, they hit their sample, and then they actually irradiate out the backside as well. What happens to healthy tissue? It's getting a very low dose again. Not necessarily all of the cells are seeing radiation, but they're in the neighborhood of cells that are. So the real question is, what's going on out in this dose area? Again, Radiation Research Society meeting has sessions about this. How do we describe the dose? We're good at describing the dose in the center, and we kill tumors, it's great, but what are we doing to the normal tissue? How do we save normal tissue from these results, from these effects? One of the ways that we've done this, and we've started looking at using particles. X-rays go in the sample and you get radiation all the way through. Particles have a unique effect that when they're going through material, they cause a little bit of damage, a little bit of damage, a little bit of damage, and then right as they're about to stop, the damage spikes up and it all comes down again. So you're able to actually program, by picking the energy, you can actually program the particles to stop where you want them to. So you have one trap coming in, very low damage, low damage, low damage, and then all the damage right there in the tumor. It's great, you can kill tumors, that's great, but something that hasn't been completely worked out yet is, okay, we're killing tumors, patients are living, that's great, but what about the healthy cells? What's going on in this low dose region, and not only that, what's going on in the cells that are right next to that low dose region? Again, the bystander effect. You don't have to directly hit them to see an effect. This is, again, Radiation Research Society actually had 2 sessions on this, on exactly trying to sort out what's going on, how do we prescribe dose, how do we prescribe risk,